The overall goal of this procedure is to perform two different mutagenesis protocols that are commonly used to generate mutants in nematodes, such as Caenorhabditis elegans and Pristionchus pacificus. Mutagenesis is a powerful tool to uncover the function of genes. This is accomplished by first preparing cultures with large numbers of worms at the last larval stage before adulthood, with which the mutagenesis will be performed. The next step is to mutagenize the worms using two alternative procedures: ethylmethane sulfonate treatment, or sorlin mutagenesis in conjunction with ultraviolet radiation. The third step of the procedure is to transfer healthy-looking worms to agar plates freshly seeded with bacteria, which is the food source. The final step of the procedure is to screen for mutant phenotypes of interest in the first, second, or third generation.
Hi, I'm Jyotiska Chaudhuri from the lab of Dr. Pires-daSilva in the Department of Biology at the University of Texas at Arlington. The TMP UV and the EMS mutagenesis procedures help answer key questions in the field of genetic research, such as which genes are involved in biological processes that has important role to play during development.
Hi, my name is Manish Parihar. Though the mutagenesis methods demonstrated will be for Pristionchus pacificus, they can be very commonly employed for other experimental models like the nematode C. elegans and even vertebrates like zebrafish. So let's get started.
Our pick for transferring worms from one location to another is made from 30-gauge 90% platinum and 10% iridium wire, although some researchers may prefer slightly different metal compositions. To make a pick, a pasture pipette is also used to hold the pick.